Welcome to this lesson on the genetic code and triplet codons. The genetic code is the set of rules by which information encoded in genetic material is translated into proteins by living cells. Understanding how 3-base sequences code for amino acids is fundamental to comprehending molecular biology and protein synthesis. Let's explore how this elegant system works. A codon is a sequence of 3 nucleotides in messenger RNA that specifies a particular amino acid or stop signal during protein synthesis. Think of codons as the words in the genetic language. Just as words in English are made up of letters, codons are made up of nucleotides. Each codon acts as a specific instruction that tells the cellular machinery which amino acid to add next when building a protein. RNA contains 4 different nucleotide bases: adenine, uracil, guanine, and cytosine. These are commonly abbreviated as A, U, G, and C. Unlike DNA, which contains thymine, RNA uses uracil instead. These 4 bases are the building blocks of all codons. By combining these 4 bases in groups of 3, cells can create a complex and precise coding system for protein synthesis. Each codon consists of 3 consecutive RNA bases, which is why we call them triplets. For example, the sequence adenine-uracil-guanine forms 1 complete codon. The triplet structure is crucial because it provides enough combinations to code for all 20 amino acids used in proteins. If codons were only 2 bases long, there would only be 16 possible combinations, which wouldn't be enough. With 4 RNA bases and 3 positions in each codon, we can calculate the total number of possible codons. 4 bases times 4 bases times 4 bases equals 64 possible codons. This mathematical relationship gives us 64 unique 3-letter combinations. This is more than enough to code for the 20 standard amino acids, which leads to an interesting property called degeneracy that we'll discuss shortly. Let's look at some specific examples. The codon AUG codes for the amino acid methionine and also serves as the start signal for protein synthesis. UUU codes for phenylalanine. GCA codes for alanine. Interestingly, some codons like UAA don't code for any amino acid at all. Instead, they serve as stop signals that tell the ribosome that protein synthesis is complete. The genetic code has 3 important properties. First, it is universal, meaning almost all organisms use the same code, from bacteria to humans. Second, the code is degenerate, which means multiple codons can code for the same amino acid. This provides a buffer against mutations. Third, the code is non-overlapping, meaning each nucleotide belongs to only 1 codon. The reading frame moves 3 bases at a time without skipping or sharing nucleotides between codons. To summarize, triplet codons are 3 nucleotide sequences that serve as the fundamental units of the genetic code. They translate messenger RNA into amino acids during protein synthesis. Understanding codons is essential for comprehending how genetic information flows from DNA to proteins. This elegant system allows cells to store, transmit, and express the information needed to build all the proteins required for life.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.